where do the microbe attach to the host cells? We know that there are some receptor molecule on host cells, and this receptor molecule typically are glycoprotein containing sugar molecule such as mannose and galactose. This receptor molecule is present not for the benefit of the infectious agents, but they have specific functions in the life of the cells. The receptor molecule present only in certain cells, which are then uniquely susceptible to infection, and also specific for certain pathogens. For example, the Neisseria gonorrhoeae has adenosine on its fimbriae that adhere to cells lining the urethra and vagina of human. When the disease is produced, there are stages that can happen. First is the incubation period. This is the time started from the exposure of the infectious agent until the appearance of the first symptoms. There is no sign or symptoms in the incubation period. After that, there is a prodromal period with very unspecific symptoms, and then the illness with the specific symptoms and more severe symptoms, and after that there is declining, the symptoms are declining, and then convalescence, when there is no sign or symptoms appear. During all those periods, the microbes can be excreted from the body through the portal exit. When it is excreted, it can be transmitted to other human beings, and there are some transmission mode as we see in this slide. Some microbes can be transmitted directly to other human beings, but also can be transmitted through vectors or reservoir. The vector is, for example, mosquito, and reservoir are mostly animals which may cause the zoonotic diseases. This slide shows you the microorganisms or the pathogens which can be transmitted through milk from human or cow. The same process occurred in the pathogenesis of viral infection. The virus should enter the host, contact and enter susceptible cells, replicate within the cells, release from the host cells, and then there is interaction with the host immune response, and this interaction will produce three outcomes: cleared from the body of the host, establish a persistent infection, or even kill the host. And after that, the microbes or the virus will be shed back into the environment. This slide tells you how the virus enters the body, and then attach to the host cells. Adsorption or attachment is a process of penetrates of host cell to gain access to the cell's replicative machinery, and this will happen if the virus have a specific protein ligand. There are two kinds of viruses, enveloped virus and naked virus. The enveloped virus will use spikers to attach to host cells. The spikes is the viral protein that protrude from their membrane. And the ligands of the naked virus is part of their capsid protein. As I told you before that the interaction between virus and the immune cells will produce three kind condition. The first one is if the immune system, especially in this case is the cellular immune response, successfully kill the virus, then the entry of the virus will not produce any disease. As we know that virus is an intracellular microorganism, so it must be in the cell. When the response immune works successfully through the cellular immune response or the natural killer cells, this process will kill or will cause the cell death, and it causes also the death of the viruses live in the cells. But if the response immune failed to kill the infected cell, and neither T cell nor NK cell can recognize virus infected cell expressing fake MHC class 1 protein, then the cell will survive and also the virus will survive. Binding of a virus to its receptor on the host cell surface result in penetration of the cell or the delivery of virus nucleic acid to the cytoplasm of the cells. To be able to produce a disease, the virus must enter the cells, and there are three ways how they enter the cells. First is they directly enter the cells. The second one, the fusion of the viral envelope with the cell membrane, and then subsequent uncoating. And the third one is the endocytosis and then release of the nucleic acid from the capsid or uncoating. After they enter the cells, it will replicate, and after replication they will release from the host cell, and there are two mechanisms of release. First is very dramatic and result in relatively large numbers of virions leaving the host cell at the same time and host cell death, and the second one is through budding or blabbing. There are two kinds of virus-host interaction. First is the cytopathic viruses, and the second one is the non-cytopathic viruses. Cytopathic viruses will ultimately kill the host cell, and the result is often local necrosis. And non-cytopathic viruses will not immediately produce cell death and result in latent or persistent infection. There are some viruses known to be the cause of cancer. In the normal condition, division of cells in a mature multicellular animal is under strict genetic control. And in the neoplasm, the phenomenon is uncontrolled cell division. The cells undergoing neoplasia are said to be neoplastic, and a mass of neoplastic cell is a tumor. And there are two kinds of tumor, benign and malignant. This slide tells you about the theory of the introduction of cancer in human. The theory suggests that more than one hit to the DNA, that is, any change or mutation, whether caused by a virus, is shown here in the second part of the figure. The yellow one is the insertion of the virus, or various physical or chemical agents is required to induce cancer. Viruses cause 20 until 25% of human cancer in several ways. They carry copies of oncogenes as part of their genome, promote oncogenes already present in the host, or interfere with normal tumor repression when they insert into repressor genes. So in summary, infectious diseases are produced through the imbalance of hosts, agents, and environment. There are some factors of hosts and some factors of agents that contributes to the production of the disease. That's all about the basic concept of infection, and I hope you will learn more by yourself. And if there is something that you don't understand, you may contact me or we can discuss it together.